So hello, everyone. Thank you for joining us today for our second session of the CSMB virtual seminar series. We're very glad that you could make it. So today we have two very awesome speakers that we're going to be hearing from. But first, I just want to pass it over to Dr. Downey to give you guys a brief overview of the CSMB conference that's going to be happening later this year.
Okay, hi everybody. Hopefully you can see my screen here. And so I'm just here to tell you about the CSMB meeting really quickly. This is happening May 30th to June 2nd at University of Ottawa. Metabolic regulation of cell signaling, but it's very broad in nature. So if you're doing any excellent work in molecular biosciences, please consider attending. Most of the talks are going to be picked from abstracts, so you have a chance to show your stuff. We have a banquet at the Canadian Museum of Nature. That's going to be awesome. The website shown here. Early registration closes tomorrow, but the prices are good even beyond that. So please spread the word and consider attending the conference. Thanks very much.
All right, so just a couple things to go through before we begin. So briefly, we're going to have a trainee talk that's 15 minutes in length, and as well as a PI talk, which is 30 minutes in length. We ask the speakers to try to adhere to these times. You will be getting warnings in the chat from me. Additionally, I'm going to be muting everyone at the start of our session. So if you have any questions once the speakers are done, feel free to raise your hand or use the chat, and I will unmute you from there. As always, this is a reminder that the seminar series is put on by the CSMB, and if you'd like to sign up and become a member, there are many perks, including the conference, which we mentioned earlier. So feel free to sign up. For trainees, I believe it's only $30 now. So yeah, with that, I'm going to pass it back to our first speaker, Laura Sosa Ponce from the University of Calgary, and she is a PhD candidate. And I'm going to let her take it away.
Awesome. Thank you so much, Kathy. Here we go. Can everyone see some microscopy? Okay, awesome. Thank you so much for having me here today. I'm really excited to present to you this work. It was just accepted into the Journal of Cell Biology last week, which is a really big deal. My first authored paper. And so I'm really excited to share the story with you. It's called SIR telomere silencing depends on nuclear envelope lipids and modulates sensitivity to a lysolipid drug, which is a mouthful, but it's okay. I'll walk you through it. Just before I move on, I'd like to highlight that I'm in the Zarenberg lab at the University of Calgary in the Department of Biological Sciences, but I was co-supervised with Dr. Jennifer Kopp. So to begin, I study the nucleus and I study it in budding yeast. And now budding yeast is a really great model organism because it's genetically tractable, it's easy to work with, but it's perfect for my study because it has a really organized nucleus. So because yeast are so small, their nucleus is only about a micron across, and they have to organize a lot of genetic information in there. So they actually make use of the nuclear envelope to make it more organized. And so the nucleolus, which is the site of ribosome biogenesis and our DNA, is actually tethered to the nuclear envelope, and most of it is silenced. And then centromeres and telomeres are also tethered to the nuclear envelope, with all centromeres being clustered into one big group, and telomeres being clustered into 6 to 8 groups per cell. So the telomeres are additionally silenced, and I'll talk a little bit more about telomeres now. So telomeres are the ends of chromosomes, and it's really important to prevent them from being recognized as ends so that we don't get aberrant recombination. They shouldn't be recognized as breaks, essentially. So that means that they're typically silenced and really bound by a lot of proteins to protect the DNA end from being sensed. So RAP1 is kind of an end protection. It's not quite the equivalent of sheltering, but it does bind telomeric repeats and protects the telomere end, and it recruits the SIR complex. Now, the SIR complex stands for silent information regulator, and this is a major histone deacetylase complex in yeast. So histone deacetylases promote heterochromatin formation. And that means that we have, you remove the lysine so that the DNA interacts more closely with the nucleosomes and you get tighter packing. So now the DNA isn't accessible for polymerases or other enzymes. But what's really cool about the SIR complex is that it's not just a deacetylase, it has tethering functions in that it interacts with nuclear envelope proteins to hold the telomeres at the periphery. And they actually have interactions with each other. So that promotes telomere clustering within the same telomere and across telomeres. So you have more heterochromatin information because you have a multi-level organization, but also you have clustering. So those 6 to 8 foci that I mentioned earlier are mediated by the SIR complex. Now, something that I haven't talked about in this image yet, you might have noticed takes up a lot of space, but it's in gray, and that's the nuclear envelope. So it's in gray because even though the telomeres have been extensively studied and the proteins that bind them have been extensively studied, the nuclear envelope hasn't been studied in terms of how it affects telomere regulation. And that is a hole in the literature because the nuclear envelope does play a role. As you can see here, there are proteins that are integrated into the envelope and they're important for interactions for tethering of the telomeres. So we have reason to believe that the nuclear envelope has a functional role because there are sensors that detect membrane changes, and one such sensor is SPT23. So SPT23 is an integral membrane protein that lives in the ER and in the nuclear envelope, and it has transmembrane helices. I'm so sorry, I can't see anyone, but I just realized that you probably can't see where I'm pointing. So here's a laser pointer. I've been pointing with my mouse this whole time. Okay, sorry, we'll continue. SPT23 has transmembrane helices that are free to rotate within the nuclear envelope. And so when the membrane is unsaturated, there's lots of movement, but when the membrane becomes too saturated and the packing of the lipids becomes very tight, then they lose that ability to freely rotate and they become stuck in this conformation shown here, which makes them susceptible to ubiquitination and subsequent proteolytic cleavage. The proteolytic cleavage frees this N-terminal domain, which can then translocate into the nucleus and act as a transcription factor. And that induces expression of lipid metabolism genes to restructure the membrane lipids. So we have sensing mechanisms here that definitely have a transcriptional response, and we know that there are domains at the nuclear periphery. So we think that there could be a role for lipids in modulating the organization of the genome and having transcriptional consequences. So in order to change the nuclear envelope in a controlled way, we used metabolically stable lipid drug adelphysine. So when you look at a bilayer, it's typically shown something like this, and each individual cylinder here is a cylindrical shaped lipid, and typically that's going to be phosphatidylcholine. That's your most abundant membrane lipid in most eukaryotic cells, and it has 2 acyl tails and a phosphocholine head group. So because this head group has roughly the same surface area as the tails, then you have a cylindrical shape which packs really nicely. But I used a drug called adelphysine, which has an inverted cone shape. Because it only has 1 tail, you have much less surface area at the bottom. And when that integrates into the membrane, it induces curvature because you are trying to match the hydrophobic packing, and so the membrane will turn to accommodate this. Now, adelphysine was synthesized. So rather than being able to remove this tail, which would occur with naturally occurring lipids, this has an ether bond, so the cell doesn't have the tools to remove that tail. And additionally, there's a methoxy group here, so the cell doesn't have the tools to add a lipid, an acyl tail. So now this is going to accumulate in the membranes and continue to induce aberrant curvature. And we know that this can target nuclear envelopes because previous studies with a fluorescent analog of adelphysine have shown that it accumulates in the nuclear envelope as soon as 15 minutes after treatment. So what we've done here is use adelphysine as a tool to change the lipid environment of the nuclear envelope and see what that means for genome organization. So the first question we asked was, what does adelphysine do to the nucleus? Sure, it accumulates, but you can see here that the microscopy was young. So we looked at the nuclear shape with our new fluorescent microscopy, and we saw that the sphericity of the nucleus is lost when we treat with adelphosine, and this becomes significant after 90 minutes. We get all kinds of fun shapes when we treat with adelphosine, but we noticed a particular tendency of something that kind of looks like a flare, where the membrane kind of juts out. And flares have been characterized in the past in response to changes to lipid metabolism. So that's typically associated with the nucleolus, and that made us question whether the nucleolus was being affected, especially since the nucleolus is also a nuclear envelope associated domain. So we looked at the nucleolus by NOP1 CFP, and you typically have what's called a crescent shape or a half moon shape for the nucleolus. But when we treat with adelphosine, we get a compaction of the nucleolus, and particularly a spherical kind of shape that tends to associate with that flare. So indeed, we are getting the nucleolar associated nuclear flare, as well as other changes, aberrant curvature. So the nuclear envelope is changing. Great, that's what we wanted. Now, what does that mean for the organization of the genome, particularly telomeres? So the first thing we looked at was tethering. We did expect the tethering to change, but surprisingly, it didn't. You can see here, this bright focus is one telomere that has a lac operon array integrated into it, and then we express lac IGFP. And so we get one bright dot where the telomere is localized, and these telomeres continue to associate with the envelope despite the deformation, and we saw this for 2 independent telomeres. So this was surprising, but we persevered, and we looked at telomere clustering, so the association between telomeres. And remember, RAP1 has a high affinity for telomeres, so we used RAP1 GFP as a marker for telomeres. Normally you have 6 to 8 foci, and we saw that here, but when you treat with adelphosine, we actually had an increase in foci. And that means that clustering is decreasing. So more dots, less associations between telomeres. And to look at that more closely, we did CHIP, or chromatin immunoprecipitation, of MYC tagged RAP1, and we saw a decrease in RAP1 binding to telomeres, although this didn't reach statistical significance. And we also did Western blots of the RAP1, and we saw that RAP1 levels were preserved, where there isn't a degradation of RAP1 in response to adelphosine, and the association with the membrane fraction of RAP1 is preserved, which is consistent with what we saw with the telomere tethering. So the decrease in RAP1 levels at telomeres is not due to a decrease in protein levels, and the clustering decrease is maybe what's causing us to see a change in RAP1 pull down from the telomeres. So since we know that the major player in clustering is the SIR complex, we next looked at SIR localization, and we did this by SIR4 GFP. And we actually found a decrease in SIR4 foci, which was almost significant. So it would suggest that the SIR is actually leaving the telomeres, and we looked at this then by CHIP, and we saw that indeed it is leaving telomeres, so we're getting less SIR4 recovery from 3 independent telomeres. So the SIR complex, remember, is important for tethering, clustering, and silencing. So our next question was, does this change reflect a change in silencing at telomeres? So we performed RNA sequencing, and this is a volcano plot showing the change of genes upregulated in response to adelphosine. And in green, we have subtelomeric genes. So we actually had a strong enrichment of subtelomeric genes, supporting the idea that SIR is leaving telomeres and transcription of subtelomeric genes is induced. What I didn't mention before is that RAP1 is also a transcription factor. And so we were curious if the change in RAP1 reflected a change in transcription as well. And we found a strong downregulation of ribosomal protein genes, which is one of the main targets of RAP1. So it looks like these telomeric factors are changing the transcription in the euchromatin as well. And finally, SPT23, which is the membrane sensor, was also enriched in both up and down regulated genes affected by adelphosine. And since we know that SPT23 is cleaved, our question was then, can we detect its activation in response to adelphysine? And in fact, when control cells, we have this membrane signal, but upon treatment with adelphysine, we have a strong translocation into the nucleus. We really lose that membrane signal. And importantly, when we treat with MMS, which is a genotoxic substance, we don't have that translocation. So this is a specific response to lipid stress. So to try to understand what's happening here, one of the major targets of SPT23 is oleic acid. So actually SPT23 is essential because of its regulation of OLE1, which is the only lipid desaturase in yeast. So if you don't have OLE1, you only have desaturated lipids, which pack really tightly and aren't really good for membrane fluidity. So you need to have some desaturation in your membrane. But we think that the induction of OLE1 by by SPT23 is causing too much desaturation, particularly because the cell is not able to metabolize adelphysine, so that it becomes additionally toxic. And in fact, in the paper, we show that oleic acid increases when we treat with adelphysine. So I hope I gave you a taste of the paper and that you'll be interested, and hopefully we can have on more information coming soon. So our model, based on what I've shown you, is that the nucleus is highly organized, but when you treat with adelphysine, you get a translocation of SPT23 into the nucleus, and the physical burden of adelphysine combined with the lipid metabolism changes induced by SPT23 causes a deformation of the nuclear envelope. And this will then have physical effects on the organization of the telomeres and the nucleolus. The big messages here are that lipid homeostasis is important for genome organization, and we're hoping to characterize this in the future with non-chemical means, so lipid metabolism mutants or directed enzyme activity. Also, lipid integrity is important for gene expression. We already knew about the sensors like SPT23, but we would really like to characterize the telomere response and see if that's a stress response, a general response to lipid changes. And finally, we saw that telomere clustering but not anchoring was affected, which made us really curious about the lateral movement of telomeres at the envelope. And we're looking for some collaborations in modeling now to investigate that further. So thank you so much for your attention and your time. And thank you to my labs, Dr. Jennifer Cobb and Dr. Vanina Sarenberg, and all my lab mates. And I'd be happy to take questions now.
Excellent. Great talk, Laura. Thank you for that. So now we'd be happy to welcome any questions. I see already that Mike is going to start us off. Go ahead.
Hi, Laura, that was a really nice talk, really clear and some nice data. And so I'm wondering if you're getting loss of the SIR complex from telomeres, are you getting more of it going to the rDNA locus? And do you see effects on the rDNA locus, such as, I guess you would predict in that case, less recombination between rDNA repeats?
Okay. That's a great question, because the TSR does partition between the telomeres and the rDNA. We see more silencing of the euchromatin, kind of like what's been shown in the literature when the tethering mutants have been deleted, but we actually didn't see changes in rDNA. So when we mutate copy number, it doesn't have any effects on adelphysine sensitivity. And when we looked at transcription of the rDNA, we didn't see any changes. What we do see is changes in ribosomal proteins and the compaction. So my current theory is based on a recent paper that showed that transcription is maintained, but it's actually held. So in order to maintain nucleolar integrity, the rRNA is actually an important part of the phase separation. So the transcription continues to occur, but the rRNA is not then translated or exported. So it's just held there. That's what I think is happening. But then it collapses because of the ribosomal protein lack of transcription. Does that make sense?
Yeah, thanks very much.
All right, if you guys have any questions, feel free to raise your hand or put them in the chat. All right, if there are no other questions, then I think we will be passing it on to our next speaker. So I'd like to welcome Dr. Wei Zhang, and he's an assistant professor at the University of Guelph. And take it away.
Okay, sounds good. Let me see the sharing. Okay. Yeah, haven't excuse me, haven't really pronounced share stories a lot. I did, was thanks to Dr. Mike Downey, I was presented my work there. So Mike, excuse me, a lot of the information here are probably you're already seeing it. Part of the reason I think I think most of the audience here are PhD or postdoc fellows, most of them. So I hope the story can give you some interest because this is, I would say, roughly, I think much less people are working on it. Doesn't mean much, but I think a lot of the sort of new things are done in some other field, especially something related to this. If your interests are related to PROTAC, some of the more small molecule based. So my work is about engineering the protein-protein interactions. I think on this part, probably most of you are interested, in terms of which proteins are interested and what are the reasons, right? And the focus of my work is to targeting the ubiquitin signal. That's all. And if you are interested, there are a few other things we are working on. So you can check out my lab if you're interested. Okay. So in terms of the outline, I just gave a couple of slides, just mention kind of more brief, it's a larger aspect, why we are doing this, right? Why we are manipulating these protein-protein interactions. That's all. Now, second part, we'll use about 15 minutes, talk about something I think is interesting, but again, it is roughly done quite some time ago. Now, the third part is more related to what my lab is working on in terms of kind of going one step further to relate it to the, sorry, I forgot to put the laser pointer. Yeah, so in terms of the targeted protein degrader, which is related to the PROTAC I mentioned at the beginning. Okay, so this is a very old, picture, which is took from some work that finished long time ago. But I, no need to look at the details. But something I just want to mention is the cellular functions are conducted through a lot of more protein-protein interactions, which are not surprising because, like I mentioned, right, a lot of you are probably working on the protein-protein interactions, and some of you are at least, interested in it. Now, the only thing I want to mention is there are a lot of those protein-protein interactions, and from my aspect is we are trying to develop something that we hope that we can use to manipulate this relatively, systematically. However, even till this point, where I started my lab about almost 5 years ago, but we didn't really do much. So we haven't really assumed to work on a lot of protein-protein interactions. Okay. Now, go back to the structure, which we probably know a little bit more. Okay. In terms of the structure dynamics of protein-protein interactions, or PPIs, makes them difficult to study and target. What that means is still a systematic way to study them. Okay. Now from A to E, depending on how much you know about the structure dynamics, as A is probably the very traditional way of we understand two proteins bind together, right? As a B is something when they are standing alone, we probably don't know much, but now when we have either have the co-crystal structures or something else, we kind of we can understand there are more to it. Now, C and D, it's essentially involving a much smaller peptide. That's all. And here, the examples, I will not go the details. Some names, I think you definitely can recognize it very, at least even you are not studying it, you probably know it right away. And at the end, it's essentially, it's more smaller, each other in terms of the pair. Okay. So this is another aspect I just want to mention to kind of related to the question that we are working on is the big question is we want to build a synthetic biology platform. Okay, we want to target those individual protein-protein interactions. That is a goal, but very hard to do. And like I mentioned, we as a lab we didn't really do a great job. Okay, over the past few years. But I hope at the end of today's talk you can understand some aspect of it. So our strategy is, I would say, quite different from most of the people. And the strategy is engineer the protein domain itself. So what that means is we're not creating a chemical compound, right? What we're doing is still let's say the two protein domains, right, A and B, we are basically engineer them, right, either A or B, but we are creating a new molecule in order to answering that question. So something more related to what we have done in the past 5 to 10 years, because I did a postdoc in Dapsidul's lab. That is when we start to, when I started to learn more about the ubiquitination, because when I, when I was a PhD student, I haven't really done much about ubiquitination. So here is the ubiquitination, summarize the key aspect. If I think for this audience, you probably know a lot of this. But just quickly jump in that. It's just focus on this yellow color, which is ubiquitin. From a basically kind of it's a cascade, right? It's binding from E1, E2, E3, showing here. The E3 are probably what we hear about the most. I think from the previous talk we talk about ubiquitination, right? RSP5, that's in yeast, right? That is a key E3 there. Now in human, there are a lot more ring E3, right? Or HAC. I think that typically people think right now is like 700 E3 ligase, and that is not important in terms of the number, but I hope that gives you some idea of how much or how many E3s there are. Right. In terms of E2, E1, they are a much smaller group, and not to say they're important, but in terms of the targeted value, they're much smaller. Okay. Now, E3 ligase, as it can target in the substrate and to ubiquitinate it, and now the ubiquitination goes to the end, which is the DUB. It doesn't really full name here. It's basically the D-ubiquitinase, which is kind of yin and yang compared to the E3 ligase, right? The DAB can remove this ubiquitination, and the proteasome is something I'm pretty sure all of you probably heard the most. Now, the other aspect is signal alternation could be, I would say actually right now compared to 10 years ago, people knows more about the ubiquitination, and they think that ubiquitination are perhaps more different compared to just targeting it to proteasome. Okay. Now, so there are these, it's kind of something we already talked about, but something I just want to mention is, roughly speaking, you can think about the E3 ligase. I mentioned the 600, but the number can be changed. They can be think as writers, right, to put the ubiquitination there. And this can be reversed, like I mentioned, the yin and yang E3 ligase versus DAB. DABs will be a smaller group, right? They can be seen as erasers. Now, the other aspect is the readers, which will be much larger, but these ubiquitin binding domains are typically much smaller. Therefore, the specificity is much harder to evaluate compared to the E3 ligase and DABs. Okay. So what we have working on, or like I said, the ubiquitination is something I started from the DAX lab. where we generate this so-called ubiquitin variant, okay? Something showing here is not generated by me, but by Dev Sidhu's lab at that time. The first paper published is 2013. So so in this case, the background is, you can think of it's just simply one, the way that ubiquitin showing here that bind to a, let's say, a deubiquinase, okay? But you can evaluate that they essentially bind the same way. Clearly, there are differences, okay? If you check the publications, you will find the differences, but they use the same, groove or same part of the ubiquitin that to bind in this case, the USP family, which is a deubiquinase family, to bind that, the same part, okay? What's that important is that now, at that time, we can generate phage display. Unfortunately, I don't have enough time to talk about phage display, but essentially you can think of as there are these different surface regions, right, of ubiquitin that we separate into region 1, region 2, region 3, but these are at the surface, right? Then you can mutate them to generate something to bind all these proteins. And that's why they are called ubiquitin variant, right, or UBVs. So essentially new proteins, right? They are very similar to ubiquitin, but quite different in terms of the sequence. Now, phage display technology is what it used to select for different binders. Okay, there are different binders can be created. For example, the one is a phage pool. You can see that here is basically the two different ways are generated. What that means is one is in the genome, the other one is somewhere on the outside. One DNA generating two proteins, it binds something else. Now in the selection, we can use this binding phage to bind this green color, which is immobilized proteins. It can be anything, right? Any proteins that you want to select for. It can be E3 ligase or deubiquinase. So what's that done? It can be selected, amplified, and some non-bind phage will be washed away. The bind ones will be selected. Now, what it does at that time is, we can select inhibitors binding to different deubiquinase. Showing here is USP8, 21, USP2A, and OTUB1. As you can see, that the key here is that this purple are the mutated residues, and they are essentially lay, or most of them, clearly not all of them, but most of them will lay at the interface to bind this deubiquinase. The only one I just want to mention quickly to you is the work I did to select for the MERS coronavirus replication. By the way, the Sidhu lab has continued, which is, after I started in Guelph, that his lab recently has published a work for SARS-CoV-2 replication using the same idea. So in this case, just to roughly speaking, that's the ME.1 to 4 bind to the MERS. By the way, it's not the coronavirus, but just one aspect of the coronavirus, essentially a deubiquinase-like part. It's a peptide-like, but the structure is quite similar. Now, the crystal structure, this is done with a collaborator. We review some key interaction sites. What's important here, just to show you, the ME.4 is one of the binder, and one not bind, okay? This MERS coronavirus can cleave, right? This is the aspect, the NSP3C24, it can cleave it, but once you have the binder, which is an inhibitor, by the way, it will not inhibit, and the UBAA, which is basically a control, it can still bind, okay? Perhaps more important in that publication is that it can protect cells from live MERS virus infection. By the way, MERS virus will be much more harsh compared to the SARS-CoV-2. But in this case, we can, the expression of ME.4 can inhibit that. Okay, something I would say interesting, but unfortunately, in my own lab, we haven't really do much yet. Okay, so the first small story is that, okay, what can we do, or what have we already did, right? This is not a new story. Like I said, it's published back in 2017 to 18. So there are two groups of E3 ligase. There are more, okay, but typically people are more interested in the hacked E3, which is a smaller group, and then the RAIN E3, which is typically the most of the E3 that people are working on, right? By the way, the RSP5, we just heard about, that is actually a hacked E3 family, okay, in yeast, of course. Okay, in terms of the RAIN, right, at that time, we target 3 representative RAIN or U-box, right? Or U-box is something similar to RAIN, but not entirely the same way. Now, the UB4B is one P-Sable. You probably heard of the Sable, but the P means phosphorylated. Okay, XIAP is the RAIN, but it is actually a dimer. So there are 3 different types, and we generate this, this UBV using the same story, generate that. And I will not have time to show you the E4B, which is bind to the UB. E4B is part of the U-box, but rather focus on the RAIN, 2 RAIN E3 family. Now the first one, UB that bind to the phosphorylated Sable. It's interesting because, as you can see here, the control, which are no UBV, it's a very short time because they change relatively quickly. And the UB74 is again like control. But once it binds the UBV that target this phosphorylated Sable, as you can see, it's essentially stop the function. Okay, the same way that the UB474, you can see the auto-ubiquitination of the Sable, but the UBV was there, it's inhibit. Okay, it's essentially something related to the inhibition of E3. Now, regarding where it binds, it's essentially bind to the E2 binding site. It's something that are typically have been shown for many different E3s. Okay, for the hacked E3s, we have some inhibitors. Typically, they bind to the E2 binding site. Okay, but it's quite interesting. Traditionally, before we published this, I think nobody published any work in terms of ubiquitin combined there. Although there are a few structural biology labs that can find that in their own, kind of unpublished work. I think there is possibility, but nobody published that. Anyway, the UBV bind to the phosphorylated Sable. Okay, this is a real structure, by the way, not modeled, but it bind to somewhere this phosphorylated tyrosine, which is, by the way, we don't have time, but essentially for Sable, the phosphorylation of this residue kind of changed the residue from the non-active and active way. Okay. Now, perhaps what's more interesting is that this binding to the XR, it's binding to the XIAP, it's actually activate. Okay, something perhaps you will remember that here, right, compared to the control, it's kind of stop, right? But in this case, compared to the control, it actually goes faster, right? Now, what's more interesting is that, by the way, here are probably too much structures, but the real structure is somewhere in the middle, where, because if you remember that we talk about XR, A and B, because it's not a monomer, right? It's a dimer. In this case, the UBV on the bottom also form a dimer, okay? This is the real one. But on the left, this E2, UBC, H5B bind to UB, but those are not real structure. Those are not the complex structure, okay? Just something I want to mention right away. So this dimer, right, UBV XR BA, that's a dimer bind to the XR A and B. Great, interesting. But what that means is that we find out that some mutations on those UBVs can make a dimer or monomer. Okay, they can change. This is the function, right? Compared to UB74, some mutations essentially change it back, meaning that once you change, not all of them, but some of them, especially the A10G, for example, showing here, is probably the only one that it change the dimer or monomer. Sorry, I don't have enough time to show you all the details, but essentially what that means is the dimer monomer change for some mutations are related to the function. However, for this one, it's harder to know because this has some other functions in terms of the binding. But later on, with this is a structure paper with the Danny lab, we actually figure out, and some other labs that are collaborate at that time with Dave Sidhu's lab are doing the same. For example, showing here the UBV W, that is another binding. It's combined this way. And the UBV, probably the most interesting one, the UBV XR 1 and 2, you can change the function in terms of the binding. And like I mentioned before, the two key interaction is a G10. G10, by the way, is the wild type residue on wild type ubiquitin. It mutates to V or A. It essentially make it differently. Becomes a dimer now. So just quickly summarize that. It's it's a flexibility of the ubiquitin scaffold in terms of the binding. So showing here, the glycine 10, once it's mutated, it's essentially open up because the ubiquitin, wild type ubiquitin can be very tight, but once it's mutated, it's kind of make it quite differently in terms of the dimer, especially. And I mentioned this a couple of times, which is a next generation dimeric UBV library, but we haven't really done much. But this is just some aspect I want to talk about in case you're interested, working on the ubiquitin. Okay, in the last few minutes, I just want to mention one thing is that related to ubiquitin and HACT, perhaps the most interesting thing is this possibilities that one UBV that can stimulate the activity, okay, of the ring. Okay, for HACT, I haven't really got time to talk about, but they can do the same in terms of the activation on the lab. It's basically inhibition. Why that's important is I mentioned at the very beginning that induced proximity of these are something people have done this degradation a lot in terms of the drug development. Okay, something related to this is there's a POI, so it can be anything. Okay, it's generally speaking not the target of the E3, but if you have some kind of drug, mostly are small molecules, right? You can then how about recruit the E3 ligase? Again, any E3 ligase, but not a lot of them has been used. E3 ligase to the POI, it can induce a degradation, which is very simpler methods. But if you search online, or you probably already know it, there are many different methods, and I would say 99% are working on small molecules. What we are working on is generally this UBV IP. So essentially just use UBV to see if we can recruit any E3 ligase, because by the way, Johnson mentioned there's only a handful of E3 ligase among 700 of them has been used. So there are a lot of opportunity to test which E3 ligase are possible. So in this case, we generate a library, right? We generate different linkers, again, very small. We're trying a lot more things, but something I would say for this audience are perhaps still useful, which is, there are E3, there are some target binding UBV. The only reason for this target binding UBV is that it is related to 53BP1. It's already been published, but the key thing is that this 53BP1 binding UBV that is very specific, also in a very non-canonical binding way to the Tudor domain of 53BP1. So that's why in my lab, we are using this a lot in terms of, okay, it binds to 53BP1 and doesn't really induce degradation. How about we use it to test other UBVs in our case in terms of induced degradation. Okay, so in terms of those UBV IP, we call it, it's basically are numbered differently here. Some are working, some are not, but most of them are working fine. Now the key thing is in this case there are two UBVs essentially induce a degradation of 53BP1. And what are them? They are essentially, one of them is binding to the RFWD3, the other bind to the NAT4L. So they all bind it, but they can lead to degradation of 53BP much better than others. Additionally, we are using some immunoprecipitation. The only thing I want to focus your idea on here is that with the DMSO, it can make the change very fast. But once the MG132, which is essentially disrupt the 53BP, sorry, the ubiquitination, it can reduce. So these two are the most of the important things that we are working on. So to summarize, that UBVs in our hand can recruit functional E3 ligase. And this tool can be utilized. By the way, we are among these two. I think we think NAT4L is more generalized. We are working with a different lab in our department and targeting something else, particularly for PROTAC, and this one worked better. And by the way, we are also working on an E2 enzyme that can work actually much better compared to NAT4L. We think it's much better, at least for a certain target. This is the key thing is for all of these proteins that are being used in terms of induced degradation, some of them may work better for this particular target. Some of them may work differently for other targets. Okay, so right now in my lab, we are doing this a lot. Essentially, we are trying to leverage different UBV IP to degrade other intracellular target, and we are exploring many other. We hope they will work, but right now the most working one, including one of the NAT4L, part of the reason is, by the way, that UBV that we find out is actually an activator of NAT4L. And other E2s, like I mentioned, is hopefully useful. And we haven't really done this a lot. Part of the reason is just the skill in my lab, but we want to do more small molecules in this case to hopefully they can be used working much better. Okay, so in the end, just want to thank you all my lab members, all the collaborators, and the funding agencies. Thank you so much. Thank you.
All right, thank you for that great talk, Dr. Zhang.
Yeah.
And now we'd be happy to take any questions. Feel free to raise your hand or type in the chat.
Okay.
Mike, go ahead.
Hi, Wei. Great
Yeah.
stuff as usual, and great to see you again.
Yeah.
Have you ever played around with, or do you think you could play around with the type of E2 or E3 used in PROTACs to change the linkage? Like if you did K63 or K11 or things like this, you could direct it to different fates or test the idea of whether this is going to direct the same substrate to degradation via the proteasome.
Oh yeah, that's definitely possible. That's definitely possible. It's basically something we want to do, but we haven't really done much. Especially, you mentioned, right? You mentioned those few different linkage. That will make things much harder, right?
Yeah.
Because like you mentioned, right? I'm sure everybody knows that K48, right, is the main linkage. But once you talk about others, it kind of expands the phenomenon to a great deal. Yeah, we haven't really done much, but that's a great point. Yeah.
Great.
Great. Are there any other questions? Okay. I can't seem to see any in the chat.
Okay.
So thank you so much, Dr. Zhang.
Okay.
With that, I'd just like to remind everyone about next month's meeting. If you'd like to join, feel free to sign up with the Zoom link over here. And once more, we will have a trainee and PI speaker. So thank you, everyone, and have a great day.
Okay. Take care, everyone. Yeah, take care, Mike.
Thank
All the
you.
best.